the Admiralty research ship Newton sails from Plymouth Sound, bound for the northeast Atlantic. Several days later, an RAF Nimrod prepares to rendezvous with her, 200 miles from land. In the same area, scientists of the Natural Environment Research Council have used a submersible to investigate the seafloor. The focus of these activities is a deep trough, and beyond it a huge submerged plateau. At its northern end, a rocky islet only 20 meters high breaks the surface.
Everyone knows the name of Rockall from the BBC weather forecasts. But to most people, it was just a name, till survey ship HMS Vidal annexed it to Britain in the name of the Queen. Vidal's helicopter takes a landing party to the tiny Atlantic island, 250 miles west of the Hebrides. Nothing lives here but seabirds, and this is only the 5th recorded human landing. But Rockall is in line with Britain's new rocket range, and since no one else claims it, it's safer in British hands.
This was news in 1955. Although the rock itself remains British territory, 3 other nations now claim rights over parts of the surrounding seabed.
Time for the shipping forecast, issued by the Meteorological Office at 23:43.
On Newton's first day out, the weather satellite image shows an area of heavy cloud associated with a depression approaching from the west. The forecast is northerly winds for 6 to 7. But what that will actually mean in terms of sea state can best be judged when the time comes.
Well, we are into our second day of the passage north, and as you can see, Newton is struggling into a northerly gale force 8. And owing to the sea state being picked up, the vessel has had to be slowed down. This means, of course, that we will now be late getting into the trials area.
This chart covers the whole of the passage, from Plymouth, 700 miles northwest, to the Rockall Bank. And these are the areas within which Newton will be working. Sandra Holt is the scientist in charge of this survey.
The Rockall Bank has an area of 10,000 square miles, covered by less than a few hundred meters of water. My research concerns its effect on the circulation and mixing in the surrounding area. The general drift of the North Atlantic current is northeasterly, but the presence of the bank disturbs this flow, resulting in a clockwise circulation around the bank and generation of eddies on the flanks. All this causes water to be displaced vertically, and we're looking for examples of this.
This underwater glider, called Seesaw, is used to measure properties of the water as it's towed behind a ship.
Right, take it away.
Seesaw is deployed from an A-frame at the stern. It's designed to follow a regular undulating path between about 10 and 250 meters depth. A hydraulic motor, driven by a propeller at the rear, controls the attitude of the wings. The hull contains sensors which measure pressure, temperature, conductivity, and chlorophyll concentration. The towing cable, faired to prevent vibration, conveys this information back to the ship electronically. The data from Seesaw are displayed in real time on a screen in the ship's laboratory. Here they're shown speeded up 30 times. The black curve on the left represents depth, and the curve on the right is temperature. Temperature decreases with increasing depth. Salinity is in the middle. Once in the water, Seesaw can be towed for days on end along predetermined tracks at a minimum of 7 knots. These shapes warn other vessels that Newton has a restricted maneuvering ability when towing. All this requires careful navigation. Traditional methods of plotting the ship's position on a chart with dividers and parallel rule are still essential, but there's a much handier way of displaying Newton's position. This video plotter enables Newton's track to be displayed continuously by combining position data, obtained from a radio or satellite navigation system, with chart information stored on a magnetic disk. Her present position is represented by the green square at the end of the dotted track. She's approaching Rockall from the southeast.
go ahead.
Maintaining a precise course while towing scientific equipment is just one of the requirements of a research vessel. Another is the ability to maneuver on station so that cables stay clear of the ship when deploying instruments over the side. CTD probes measure conductivity, temperature and depth from top to bottom of the water column. They're slower to use than seesaw, but can go much deeper. They're useful over rugged topography where seesaw would be at risk. Oceanographers also need to know where they are in relation to bottom topography. This precision depth recorder plots the water depth as Newton steams westward from the Rockall trough onto the Rockall bank. The seabed shallows from over 1,000 meters on the left to about 250 meters on the right, with a maximum gradient of 1 in 3 between.
Here we are at Rockall, having completed the southern half of the survey. The weather is very good for this location. We're now going to proceed up to the northern half and complete the survey.
Okay, level, half a mile to run. Wind here, please.
Even from the air, it's possible to make oceanographic measurements. Just looking through the window gives an experienced pilot a good idea of sea state, and aerial bathythermographs can be dropped to investigate the waters beneath the surface.
Okay, crew from Denham, stand by, Bathy, channel 16, in position 5728 north, 1256 west. Channel 16, now, boys gone.
On contact with the water, the instrument splits in two. The weighted temperature sensor falls to the bottom, sending by wire a signal up to the floating transmitter. On board the aircraft, temperature and depth are displayed as the probe falls to the sea floor. Here you can see evidence of a thermocline below about 30 meters. The printer produces a permanent record for examination back at base. Meanwhile, Newton was doing all that was asked of her. The diesel-electric propulsion system causes very little vibration, helping to provide the stable yet maneuverable platform needed for oceanographic research. The program of sampling and measurement continued 24 hours a day as she steamed back and forth gathering data. At night, the warning shapes are supplemented by lights. The scientists worked continuous watches of 6 hours on, 6 hours off throughout the survey, in all weathers.
Now the survey is nearing its end. There were times when everything seemed to go wrong at once, and all we could do was tackle the nearest thing and muddle through. But it's a good feeling at the end of a survey when you realise you've done the best you can under the circumstances. In 12 days, we covered 2,000 nautical miles and collected a vast amount of data. The detailed data interpretation must await analysis back on shore.
In the Admiralty Research Establishment at Portland, the data obtained on Newton are being plotted by computer. Here are some of the preliminary results. These vertical profiles of temperature in the top 140 meters of the water column are from two positions 15 miles apart on the southeastern slope of the Rockall bank. The one on the left is in water with a total depth of about 800 meters and shows a weak thermocline at around 50 meters. The one on the right shows a much stronger thermocline in a total water depth of about 400 meters. By combining all the temperature data between these positions, we can obtain a cross-section showing how the depth of the isotherms changes as we approach the top of the bank. Here, in the area corresponding to the first profile, the 10-degree isotherm is at about 140 meters, but as we move north, the isotherms rise abruptly and the 10-degree isotherm is only 60 meters deep, in the area of the second profile. Eventually, a complete picture of the temperature structure can be built up. This may help to answer some questions about biological productivity in the area. Satellite images during the summer show the Rockall bank as a dark patch against a more reflective background caused by the presence of microscopic phytoplankton, with shells made of calcium carbonate. These blooms of coccolithophores contain millions of cells, like this one, in every litre of water. Perhaps their growth is stimulated by nutrients brought towards the surface in the cooler water from below. After death, their broken skeletons become part of the ocean floor sediment.
Intuitively, you wouldn't expect such surface events to have much influence on the deep sea floor far below, because these tiny organisms would take so long to sink through the water column that any seasonality would not be reflected by the time they reach the bottom. Indeed, during our work in the Porcupine Sea Bight to the southwest of Ireland, we had no evidence of this for some time. We'd used an epibenthic sledge, a frame with nets on the back, which is dragged across the seafloor to collect animals living on and within the sediment. And on the frame of this net, we routinely used a time-lapse underwater camera directed forwards and downwards to photograph the seafloor ahead of the net. During our first few cruises during the winter months of the year, these produced quite clear pictures of the bottom with evidence of movements of benthic animals, depressions, mounds and so on, but nothing much else. And then in the early 1980s, we had a number of cruises at different times of the year, in May and July. And the photographs obtained now, at this time, were quite different from those which we'd obtained earlier. When we look carefully at these, the depressions were all filled with an amorphous, dark-coloured, flocculent sort of gunge. Sometimes this material was so abundant that it made the pictures look as though they were out of focus and even hid some fairly large organisms. What on earth was this material, we thought? Where did it come from? Clearly, between the winter and the summer, something very dramatic was happening on the deep sea floor. But what?
This suggestion that the appearance of the deep sea bed may change seasonally ran quite contrary to what we then believed. One obvious way to examine this further was to photograph in time-lapse one patch of seabed over a long period of time. This is the equipment we use to do this. This is a deep-sea camera which photographs a patch of seabed such as this. Of course, there's no light down there, so the area is illuminated by the flashlight. In order to get the rig down to the bottom and to hold it there, we use this massive iron tripod here. And when we want to bring the equipment back up to the seabed, this ballast weight is left behind after the trigger here is activated from the research ship several kilometers above. The buoyancy spheres then drag it back up to the surface where it's recovered. So much for the technology. What did we actually find out? In May 1983, we dropped this rig into 4,000 meters of water on the abyssal plain just next to the United Kingdom. We set the camera to photograph every 8 hours and left it on the seabed in this rather hostile environment until our return several months later. We're looking at 2 square meters of seabed, and to give a feeling of scale, the mound on the right is about 18 centimeters across. As you can see, there is little change in the appearance of the seabed during May and most of June. Some animal tracks are formed, particularly at the bottom of the frame, while others degrade, but the scene is largely unchanged. In the latter part of June, however, you can see dark blobs of material of up to a centimeter in diameter arriving on the seabed. These move about and change in appearance, but by mid-July, the seabed is covered with a thick layer of detritus. Unfortunately, the film ran out in mid-August, so we do not see a complete return to the winter situation. So what, you may ask, is the relevance of these observations to the oceanic environment as a whole? What we've demonstrated is that although the top and bottom of the ocean are separated by several kilometers, there is in fact a very close coupling between these two environments. Processes occurring in the top 50 meters have a direct and large effect on the rest of the water column beneath it. This deduction was only possible when we were able to determine that the fluffy detrital layer was fresh phytoplankton with an oceanic species composition.
What I'm looking at here is a frozen sediment core collected at a water depth of around 4.5 kilometers in the northeast Atlantic. It's been extruded from its coring tube. If we thaw parts of this core and examine them carefully under the microscope, we find that the sediment will contain hundreds or even thousands of tiny organisms. I find that most of these organisms live in the top 1 or 2 centimetres of the sediment. In cores collected during the summer months, we find that the sediment surface is sometimes overlain by a greenish-brown layer of phytodetritus. The colour of this material suggests that it contains phytoplankton remains. However, in order to examine it in more detail, we have to look at it under the microscope. The detritus consists of a loose matrix containing shells of various single-cell planktonic organisms. Looking in detail at different parts of this slide, we can pick out delicate hexagonal structures which are the skeletons of silicoflagellates, about 30 microns across. Over here is a bell-like shell of a tintinid, about 80 microns long, or just visible here, the coiled, multi-chambered shell of a juvenile planktonic foraminiferan, about 60 microns across. The shells of these planktonic protozoans have been transported several kilometres down from the sunlit surface waters where they lived. However, the phytodetritus also provides food and a living space for deep-sea organisms. The most important of these are undoubtedly the bacteria, which invade the phytodetritus in large numbers. Larger and more obvious, although much less abundant, are benthic foraminifera. These bottom-dwelling protozoans are rather like amoebae with shells. This view shows a specimen with a coiled, multi-chambered shell embedded in the detritus. Evidence that the foraminifera feed on this detritus is provided by the green colour of the protoplasm, clearly visible through the transparent shell of the specimen which has been extracted from the detritus. So here we have plant-eating organisms living in a quite unexpected place, 4 kilometres down on the floor of the Atlantic Ocean, and providing a dramatic biological link between the surface waters and the deep-sea floor. What happens to the phytodetritus? Well, the bacteria undoubtedly play a major role in degrading it. Large surface-feeding organisms such as sea cucumbers eat it, and other organisms such as worms mix it down into the sediment. The remnants may be scattered by currents. Whatever its fate, the phytodetritus disappears from the sediment surface by the end of the summer. So the benthic organisms which make use of it have to wait until the following spring before another food bonanza descends through 4.5 kilometres of water to the deep-sea floor.
The population biology of some of these organisms is being studied at the laboratory of the Scottish Marine Biological Association near Oban.
What I've been trying to do over the past few years is to take samples from one position in the Rockall Trough at about 3 kilometres depth in order to follow the breeding and growth of individual small species of these small benthic animals, such as this small, oddly shaped sea urchin. This is a 2 centimetre long specimen, and I would guess it to be about 3 or 4 years old. Well, how would I know this? Well, when we look closely at its hard outer covering, and heat-treated especially, this is what we see. No, it's not a tortoise shell, but what was fascinating to find was that we can see a ring-like growth pattern in the individual plates. In this separated plate, we can see clearly how closely this ring-like pattern resembles that in the trunk of a tree. If we look closer still at the skeleton of this species with a scanning electron microscope, we can see the telltale differences in the density of this spongy-looking fine structure, showing that this little echinoderm grows in distinct bursts. When we looked at some even smaller animals, such as this tiny bivalve from the same sample, we can see rather similar rings that, when I saw them for the first time, reminded me instantly of the pattern of growth rings that one can see on the shells from the beach. So it seems likely that in the deep sea, their cousins show a similarly discontinuous pattern in growth. And if we assume that the spacing of these growth rings has an annual period, then by counting the numbers of rings, we can make an estimate of the age of individual animals. And all this makes it difficult to sustain the classical view of great constancy that supposedly characterizes the deep-sea environment.
Calanus is the Scottish Marine Biological Association's coastal research vessel. Her sampling gear includes a lightweight trawl which is towed along the bottom by long warps attached to the boat's winches.
Up a bit, George. This is a typical research catch from a depth of about 50 metres in an inshore area. What have we got here? Well, we've got dogfish, skate, plaice, various other smaller flatfish. But as you see, most of the catch is made up of the cod-like family of fishes. And what happens when we go out to the edge of the continental slope? We still get some of these species, for example the cod, but as we go down the slope, this family is replaced by a closely related family of macrurid fishes. Here we have a small macrurid which lives at about 1,500 metres, and you can see here that it's one of the types that have a long pointed snout which is partly armoured, and it also has a long tapering tail which is typical of the macrurid family. This species is one of the round-nosed type of grenadiers, or rat-tails. It has, again, a tapering body, and this one starts in Rockall Trough at about 2,000 metres and lives down to depths of at least 5,000 metres.
Calanus is not equipped for trawling at these depths, but the British research ship Challenger routinely trawls down to 3,000 metres. This is a catch from about 1,000 metres in the Rockall Trough. Unusually, it's dominated by one species of Olipocephalid, commonly called smoothheads or slickheads. These fish belong to a family closely related to the herring, but lack a swim bladder, and achieve neutral buoyancy by having very watery flesh. This large catch from 1,000 metres consists almost entirely of adult fish in spawning condition, suggesting that fish at mid-depths may have seasonal reproductive patterns.
Because of their watery flesh, these Olipocephalid fishes are unlikely to have any commercial importance. However, slope fishes are exploited in other areas of the North Atlantic, such as Canada, Greenland and Iceland. And the most common species caught is this macrurid or round-nosed grenadier. Although the resources of the Rockall Trough are not yet fully exploited, almost certainly there will come a day when fish such as this are commercially marketed. And it is important that we understand as much as possible about the biology and life history of these fish so that it can be effectively managed when the fisheries begins.
Fish are not the only potential new resource around Rockall. Major oil companies have been exploring the area for hydrocarbons. These commercial possibilities have led to 4 nations claiming rights over parts of the surrounding seabed: the UK, the Republic of Ireland, Denmark by virtue of its sovereignty over the Faroes, and finally Iceland, all on the grounds that these areas form part of their legal continental shelves. This uncertainty of ownership may inhibit future commercial developments. Irrespective of when these issues are resolved, the huge isolated mass of the Rockall Bank and the adjacent deep waters of the Rockall Trough will continue to provide oceanographers with a convenient natural laboratory in which to investigate processes occurring throughout the oceans.